Hello everyone, this is Medical Gateway, and today we're going to study a very important topic, which is translation. Translation is basically that how messenger RNA produces protein, how the protein is formed by RNA. So let's start it. Okay, in this chapter we'll also discuss different types of RNA like messenger RNA, transfer RNA, and the ribosomal RNA. Okay, so let's start. Now the first thing is that this is the ribosome. This circular shaped organelle is the ribosome. On ribosome, this pink colored chain is the messenger RNA, and then this blue colored clover leaf shaped structure is the transfer RNA, and here these are the amino acids, different amino acids, which are collectively forming a protein chain. Okay, so this one is the ribosome, one ribosome, and this one is the other ribosome. On ribosome, there is messenger RNA, which has come from the nucleus into the cytoplasm, taking the message of the DNA in the form of codon to the ribosomes for the formation of the proteins. Okay, now what happens exactly is, if you focus on the messenger RNA, it has a specific sequence AUG, GCC, GU, GU. So as discussed in the previous lecture, the sequence of the three bases will form a codon. So this one is one codon, this one is the other codon, and we have labeled here this CCC sequence as a codon. Okay, now this codon AUG will define a specific amino acid for the protein formation, and this GCC will result into formation of, will define another amino acid. So these all codons will define respectively a single amino acid, which will be attached in the form of chain, and then a protein chain is formed. Okay, now what is the function of this transfer RNA? Okay, so transfer RNA is a clover leaf shaped structure. Now it looks as it has two strands, but no, this is a single stranded structure. It starts from here, and then the strand bends here, then it bends again, then it bends again, and it is ending here. So this is a single strand which is arranged in the form of a clover leaf shape. Okay, now as the messenger RNA has a sequence of bases, this transfer RNA also has bases. For instance, there might be adenine here, guanine here, cytosine here, uracil here, and now, but we have to focus at this point of the transfer RNA chain. We can say at the base of the transfer RNA, but basically this is exactly the middle portion of the transfer RNA chain. Now this portion has three nitrogenous bases, and different transfer RNAs have different sequence of bases. Now that transfer RNA will attach on this codon, which has the nitrogenous bases complementary to this codon. So the sequence of this codon is CCC, which means all the three are cytosine. Now, as you know, cytosine is always forms bond with guanine. So if a transfer RNA has consecutive three guanine, guanine, and guanine in the middle of its chain, that will attach to this part. Okay, for instance, which transfer RNA will attach to this codon? The first one, AUG, the start codon, from where the translation is going to start. The transfer RNA which has UAC sequence in the middle will get attached here. okay, and then the transfer RNA which has C, G, G, all the counterparts of this GCC will be attached here. Now, each transfer RNA has the ability to carry a specific amino acid. Okay, for instance, this transfer RNA is carrying an amino acid which is proline. Okay, so now the DNA has the genetic code, DNA has formed the RNA, messenger RNA has brought this code in the form of codons to the ribosome in the cytoplasm, and then there the transfer RNA will see this, will read this code in the form of different codons and bring the respective amino acids from different parts of the cytoplasm to the messenger RNA, and those amino acids will be attached in the form of a chain and the protein will be formed. For instance, AUG will make alanine, then GCC will make cysteine, then UGU will make histidine, and they all will be attached in the form of a chain. And when the turn comes of this CCC, then this transfer RNA will bring the proline, and the proline will be added to the sequence of amino acids and protein is formed. Okay, so this is how exactly the messenger RNA transfers the message of DNA to the transfer RNA in the cytoplasm, and a protein chain is formed. So messenger RNA is this which is carrying the message, transfer RNA which transfers different amino acids from different places of cytoplasm and forms protein chains, and third one is the ribosomal RNA which forms the structure of the ribosome on which this whole process is taking place. Okay, now, so the point from where the translation has to begin, the code at that point is known as the start codon, and the point up to which the translation has to occur, or the point where the translation ends, is known as the stop codon. Now this sequence will define the starting point and UAU will define the stopping point, so this is known as the stop codon. So whenever the sequence is UAU, it means it is a stop codon. There are two other sequences of stop codons, UAG and UEA. So you have to remember these three sequences. These three sequences are said to be stop codons. AUG, you can remember it from AUG, from augmentation. You can, it's a mnemonic for the start codons, and for stop codons you have to remember UAU, UAG and UEA. These all are stop codons. Okay, so this whole is how the mechanism of the production of protein. Now let's discuss this in another diagram which makes it a little more relatively easier to understand the concept. So this is the diagram. Now this brown colored membrane is not the messenger RNA. This is basically the endoplasmic reticulum, and on the surface of this endoplasmic reticulum are ribosomes. This 1 ribosome, 2, 3 and 4 ribosomes on the surface of this endoplasmic reticulum. As you know, endoplasmic reticulum has two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is said to be rough because it has ribosomes on its surface. So these are ribosomes on the endoplasmic reticulum. Now just focus on this ribosome. Now this is a messenger RNA. So this messenger RNA comes here on the ribosomes with the message, with the code, with the codon, okay, for the formation of the amino acids. Now this is a transfer RNA. So this transfer RNA will carry the amino acid, and then this transfer RNA will be attached to its respective counterpart bases on the messenger RNA, on that part where, and that codon where its bases are in counterpart with the bases of the messenger RNA. So this is how the transfer RNA brings different amino acids from the cytoplasm and take them to ribosome where the messenger RNA is placed. Now what happens that this amino acid will be attached to the existing chain which is already being formed with reference to the different sequence of the codons, and the whole protein chain will be formed, and then there will be another amino acid attached, then another amino acid attached. For instance, there are around 8 to 10 amino acids already attached to this protein chain, and then a new amino acid is attached. Then after this, there will be another amino acid will be attached until a stop codon is reached. Okay, and then once a transfer RNA has done its job, that transfer RNA then will detach itself from the messenger RNA, then it goes again into the cytoplasm. Okay, and when there is stop codon is reached, then the complete chain of protein is detached from the transfer RNA, and then that protein will go into its function, whether it is a structural protein or whether it is a functional protein in the form of enzymes. They will do its functions. Okay, so this is how the protein is being formed on the ribosomes with the help of messenger RNA and the transfer RNAs. Now we have discussed a single ribosome, but there are many ribosomes here. But interestingly, all of these ribosomes, there is a single messenger RNA chain. So for instance, the messenger RNA chain starts from here. This will run from this. First of all, it will go on this ribosome, then this chain will keep moving forward, and on this ribosome, then the other one and the other one. So 3 to 10 ribosomes along with a single messenger RNA, this whole combination is known as polyribosomes. Now, what is the significance of this polyribosome? For instance, this part has generated its codon, and the transfer RNA came, and amino acid is attached to the protein. Then this sequence will move forward to the other ribosome, and then to the other ribosome, and then another ribosome. So collectively, there is formation of the same protein for which the messenger RNA has brought the message from DNA. So there will be 4 different chains of the same protein is being formed at the same time. This is the one on this ribosome, this chain is being formed. On the other ribosome, this chain is being formed. On the other ribosome, this chain, and then this chain. So at one single time, one messenger RNA is being translated into 4 different chains of amino acids to form 4 different chains of proteins, and all of these are the same proteins. Okay, so this is the significance of the polyribosome. So polyribosome is a combination of a single strand of messenger RNA along with 3 to 10 ribosomes. Okay, so now let's discuss different types of ribosomes. So one is the messenger RNA, this one, which carries the message. So it carries genetic code to cytoplasm. The other one is the ribosomal RNA. The ribosomal RNA provides structure on which the protein molecules are assembled. Just here, it provides a structure on which the protein molecules are assembled. So basically, ribosomal RNA, along with 75 different proteins, makes the ribosome. So what is the structure of ribosome? Ribosomes consist of ribosomal RNA and 75 different proteins. Then we have transfer RNA. What is the function of transfer RNA? It transports amino acids to ribosomes. Okay. Now, other than these 3 types, there are 2 other types of RNAs. One is the precursor messenger RNA. Now, from the name, you just can tell that this is a precursor of messenger RNA. So precursor messenger RNA, what is the fate of precursor messenger RNA? It will be converted into the messenger RNA. And who will convert precursor messenger RNA into messenger RNA? That is small nuclear RNA. Okay. So someone asks you, what is the function of small nuclear RNA? You will say it converts precursor messenger RNA into messenger RNA. Okay. Okay. Then we have two other types. One is microRNA. Now, what is the function of microRNA? This is very important. MicroRNA regulates gene transcription and translation. If there is an MCQ, that which of the RNAs regulates gene transcription and translation, you will say microRNA. So microRNA regulates gene transcription and translation, so that the process of transcription and translation is not uncontrolled. Just microRNA controls this whole process, regulates this whole process of transcription and translation. Now, if the microRNA, so microRNA is very important. It regulates gene transcription. It just puts a stop to the transcription and translation and regulates it. So if the microRNA is made synthetically, so the synthetic microRNA is known as small interfering RNA, silencing RNA, or short interfering RNA. From the name, it is pretty obvious that silencing RNA. Now this RNA is silencing. It puts a stop to the process of translation. Or short interfering RNA or small interfering RNA. So this RNA interferes with the process of translation, so that the translation happens in a controlled mechanism. The amount of the protein formed should be controlled. Okay. So microRNA regulates gene transcription, and its synthetic forms, which are known as short interfering RNA or small interfering RNA or silencing RNA, they all regulate gene transcription and translation, but these are the synthetic forms. Okay. Okay, so now let's discuss the regulation of gene expression, because in the last function here we discussed that they regulate gene transcription and translation. How exactly are they regulating the gene transcription? So regulation of gene expression by microRNA. Okay, so microRNA wants some importance now. So now microRNA, basically in the nucleus, it is not present in the form of microRNA. It is present in the form of primary microRNA. So there is a thing, microprocessor complex, that converts this microRNA, this primary microRNA, into pre-microRNA. And then this pre-microRNA goes into the cytoplasm, and there's an enzyme known as the dicer enzyme. This dicer enzyme converts this pre-microRNA into microRNA. So microRNA's the first form is primary microRNA, then pre-microRNA is formed, which moves to the cytoplasm from the nucleus, and then the microRNA is formed, which is converting primary microRNA into pre-microRNA, that is microprocessor complex. And what is converting this pre-microRNA into microRNA? That is dicer enzyme. Now, what this microRNA, which is finally made up to 2-3 steps, what this is going to do? This will regulate the gene expression, but how exactly it is going to regulate the gene expression? So microRNA with RNA induced silencing complex, RISC. So microRNA along with RNA induced silencing complex basically activates an enzyme which is RNase, and this RNase cleaves the RNA. Okay, we have discussed earlier that microRNA regulates gene expression. microRNA will control the production of the proteins by the translation. So microRNA can put a stop or can silence the process of transcription and translation. Okay, so how it is going to do, when it is formed in the cytoplasm, it will act in coordination with RNA-induced silencing complex. Now, this RNA-induced silencing complex will use this microRNA as a template for recognizing complementary messenger RNA, where this RNAs will act and cleave the RNA and stop or stop is not the right word, or control or regulate the process of gene expression. Okay, so our RNA-induced silencing complex is using this microRNA as a template to recognize what? The complementary messenger RNA. And then the RNAs will be activated, and that RNAs will cleave the RNA and will regulate the gene expression. Okay, so this is the whole function of microRNA. Okay, until now we have discussed the basic mechanism of the translation, and we have discussed different types of RNA, including the microRNA. Now just one point is left, and that is chemical events in the formation of protein molecule. Okay, messenger RNA has brought the message, transfer RNA has transferred the amino acids, but how exactly the different amino acids will form bond with one another? Are there any chemical events? Are there any use of ATP or any other thing involved? Or are there any other enzymes which are helping them to form the bond? So let's discuss that. So these amino acids, when they have to form bond with each other to form the protein, these amino acids will first be activated. How activated amino acids are formed? Amino acids will be converted into activated amino acids with the help of ATP. ATP will give its two high energy phosphate bonds, and amino acid will be converted into a combo of AMP and amino acid, which is basically activated amino acids. So now this AMP and amino acid will get attached with the transfer RNA, making a combination of transfer RNA and amino acid. Now this transfer RNA and amino acid combination will move towards the ribosome on the messenger RNA, where this amino acid will be attached to the other amino acids. Now this transfer RNA amino acid combo will get attached with messenger RNA, making a transfer RNA, amino acid, and a messenger RNA trio. Now this transfer RNA, amino acid, and messenger RNA will finally give amino acid to the already formed chain of protein. This amino acid will get attached to the other amino acid formed from the previous codon. And for this purpose, there is an enzyme, peptidyl transferase. Now this peptidyl transferase will come, and this will form a bond between two adjacent amino acids. But this process also wants two high energy phosphate bonds, just as the first step. So in the first step, where the amino acids get activated, are converted to activated amino acids, two high energy phosphate bonds were used. Now in this process, where the amino acids, when they are being transferred by the transfer RNA and they are present on the messenger RNA, there again ATP will come and will give its two high energy phosphate bonds, and then the amino acid will form bond with the other amino acid and form the protein chain. And this final step is facilitated by peptidyl transferase, which is an enzyme. Okay, so this is the chemical event. These are the chemical events in the formation of protein molecule. So by this, we have finished translation. We have also discussed that there are chemical events present in them. We have also discussed in this part different types of RNAs. See you in next part.